Hi everyone. My name is Anvi Sharma and I'm currently a senior in high school based in Bangalore, India. My mentor's name is Carl Anderson and my paper is titled Endocrine Disrupting Chemicals in the Gulf of Thailand, a review and identifying knowledge gaps. So first, I wanted to talk about my inspiration behind this project. So I initially started exploring endocrine disruption in wildlife because of my interest in the intersection of biochemistry and wildlife biology. And what really piqued my interest in this field was that despite the far-reaching consequences of endocrine disruption, discussions about this subject tend to focus primarily on human health, and the implications for wildlife and ecosystems are often ignored. So I wanted to shed light on these neglected aspects. During my first literature review, I discovered that a significant body of research on endocrine disruption does exist, but there is a noticeable gap in studies focusing on specific geographical locations or species native to those areas. And I was really inspired by this realization and the recent trip to Thailand, so I decided to narrow down my focus on endocrine disrupting chemicals in the Gulf of Thailand. So, coming to some information about the Gulf of Thailand, it is Thailand's largest coastal waterway and main oceanic access, and hosts prominent fisheries, tourism industries, and a petrochemical industry that accounts for 5% of the country's GDP. So, increasing anthropogenic activities and industrial activities have made this area particularly vulnerable to potential contamination from EDCs. And this poses a substantial threat to the impressive biodiversity and multiple habitats of the Gulf. It is also a risk for the human population because endocrine disrupting chemicals or EDCs have been found to be accumulated in marine life that are dietary staples for coastal communities. So coming to the first chemical that I researched about, it's microplastics, which might not be surprising to most of you. So scientifically, we define microplastics as pieces of plastics that are less than 5 millimeters in length. And their most common source of origin is degradation of larger plastic items. Not much is known about microplastics and their impacts as an endocrine disruptor on wildlife yet, but we do know that their chemical composition enables them to contain or absorb EDCs, in particular chemicals involved in plastic production. And they might have similar structures to natural hormones, allowing them to bind to hormone receptors and disrupt normal hormonal signaling, which would lead to inappropriate activation or blocking of hormones and receptors and altered physiological responses. In the case of marine organisms, they often tend to mistake microplastics as food, which causes them to ingest them and leads to bioaccumulation and biomagnification. So, microplastics have also been found in particularly sensitive habitats in Thailand and other parts of the world. So, first, mangrove ecosystems, which have a particular high sinking potential for microplastics because of their plant root systems and low energy environments. And what's concerning about this is that mangroves act as a nursery for young ocean animals, and exposure to endocrine disrupting chemicals and microplastics can lead to developmental impacts early in life, which can have an adverse effect on overall health. And coming to coral reef ecosystems, which is another area where microplastics have been detected, microplastics physically damage tissue, reduce growth and reproduction, and alter the behavior of associated coral species, having an overall negative impact on the marine ecosystem. So now focusing a little bit more on their impact on wildlife, a recent study conducted in the Upper Gulf of Thailand found that 46.9% of the sampled fish contained microplastics in their gastrointestinal tracts. And while no direct associations were found between microplastic presence and histopathological changes in fish tissues, microplastics can still act as EDCs, and this would not be necessarily detected in histopathological lesions. Another study showed that microplastics were found to be accumulated in every organ of a select population of studied catfish and may have contaminated catfish eggs, which is extremely concerning because it demonstrates how microplastics are very much present in our natural environment and wildlife. Next, BPA. So I'm sure a lot of you would be familiar with BPA because of the whole BPA free movement that has gained traction in the past century. So BPA is a chemical associated with microplastics, and it is a known endocrine disruptor. So it was initially considered to be a weak environmental estrogen, but more recent studies have demonstrated that BPA might be similar in potency to estradiol, a strong steroid version of estrogen in stimulating some cellular responses. This is a concern because BPA can influence multiple endocrine-related pathways and can have implications on genetic and epigenetic dysregulation. So these graphics show the similarities between BPA, DES, and estradiol, and also the chemical structures of associated products of BPA, which gives more insight into how the chemistry behind BPA allows it to act as an endocrine disruptor. Coming to the effects of BPA on wildlife in the Gulf of Thailand, a study investigating the effects of BPA in clown and fish found that BPA is present in significant concentrations in the Gulf of Thailand. And BPA exposure has also been shown to induce vitellogenin synthesis in fish, causing reproductive issues like reduced sperm quality and delayed ovulation. It is acutely toxic to aquatic organisms at low concentrations. High concentrations of BPA were also detected in muscle tissue samples from both coastal areas and freshwater sites. So since we know that BPA can have massive effects on the health of humans, it is entirely possible that they can have similar effects on wildlife. So more research is required to find out more about these effects and start tackling them. Moving on to phthalate esters, they're a group of chemical compounds that are also used in plastic production, and they exhibit anti-androgenic properties, which essentially means that they inhibit the action of androgens, which is critical for reproductive health. So this interference can lead to a range of reproductive issues, including altered sexual development and fertility problems in various species. So again, phthalate esters, as I mentioned earlier, is a chemical compound associated with plastics, and so is BPA, which ties back into how microplastics can be a very dangerous endocrine disrupting chemical and highlights why it's so important to tackle this issue right now. So moving on to tributylin or TBT. So tributylin mainly interacts with nuclear receptors, which alter various reproductive, developmental, and metabolic pathways, leading to significant physiological changes in exposed organisms. So one thing about TBT that has been documented extensively is its ability to cause imposex, which is a condition where female gastropods develop male sexual characteristics, and it occurs due to TBT's ability to inhibit aromatase, an enzyme crucial for converting androgens to estrogens, leading to elevated testosterone levels in females. So this results in reproductive impairment and decreased population viability among affected species, and it has also been documented in the Gulf of Thailand. In addition, TBT exposure has also been shown to cause masculinization in fish, disrupting normal sexual differentiation. And this is concerning because fish species are particularly sensitive to TBT, with endocrine type responses observed at very low concentrations. So again, it's very important that we eliminate TBT from our environment for the health of our wildlife. Finally, I will be talking about metalloestrogens, and this is the part which kind of inspired my whole paper. So these are a class of endocrine disrupting chemicals that include certain heavy metals and metalloids, which can mimic or interfere with the actions of estrogens in the body. These substances can then bind to estrogen receptors, leading to various biological responses that can disrupt normal hormonal functions. So there are various ranges of mechanisms of metalloestrogens, including estrogen receptor binding, oxidative stress, and disruption of hormonal pathways. So these impact dugongs, large marine mammals found in warm coastal waters. They very closely resemble the Florida manatees. And dugongs are known to accumulate heavy metal ions in their tissues throughout their lives, more so than many other marine mammals. And this accumulation can lead to toxic effects, particularly as these metals mm, can interfere with hormonal signaling pathways, and they may also disrupt the reproductive health of dugongs, leading to altered reproductive behaviors and physiological changes, potentially affecting the calving intervals and health of calves. And dugongs occupy the seagrass beds habitat of Thailand, and this is already going through rapid habitat degradation, which can be exacerbated by pollutions, including heavy metals. Moreover, dugongs have been already in very low numbers in Thailand, so it's very important to tackle the issue of endocrine disruption to help bring back numbers and increase the health of the population. So this brings me to the end of my presentation. Thank you so much for listening, and feel free to reach out to me on LinkedIn if you have any questions. Thank you.